This is Brain Ponderings. I'm Mark Matson, and today I'm going to talk about two evolutionarily very important bioenergetic challenges that we are all faced with, although maybe less so in modern societies, and those are the need to exert physical effort and to go extended periods without food intake. So physical exercise and intermittent fasting. There's been a lot of work looking at how these two environmental bioenergetic challenges affect health in general. And recently, there's been a lot looking at the brain as well, which I'll focus on today. And it makes a lot of sense from an evolutionary perspective that the brain evolved to function very well in food-deprived state, and that under those conditions where an animal or our human ancestors before the agricultural revolution uh, had to spend a lot of time trying to find food, that their brain had to be working well and they had to be able to spend a lot of physical exertion in that food deprived state. Now, there have been quite a few studies looking at the effects of physical exercise, mainly aerobic exercise, so running, biking, brisk walking on the brain in terms of its function and in terms of even structure. And this slide shows some data from a study done by Art Kramer's group. By the way, I had a podcast with Art Kramer. It's been quite a while ago, maybe a year and a half or more ago. So you might want to check that out if you want more on effects of exercise on the human brain. But in this study, what they did is they took older human men and women, and they divided them into two groups. One group, the control group, they had to do just daily stretching, but no aerobic exercise. And then the other group, they had to do 40 minutes of brisk walking daily, and these were supervised. So it was clear that they actually did the exercise. And then what they did is they took MRI images to look at brain structure, and they quantified the size, the volume of the hippocampus, both the left hippocampus and the right side of the brain, uh, before they started the interventions, and then at 6 months and 1 year later. And what you can see in the upper part for the hippocampus is that the individuals who were doing aerobic exercise during that year, by the end of the year, they had a significantly larger uh, hippocampus than those on the stretching, the control group. In fact, the people in the control group, there was a small reduction in the size of the hippocampus over a month. Now, remember, these are older people whose hippocampus is going to be decreasing just naturally. These are cognitively normal people. That is, they don't have dementia or any neurological disorder. Okay, and then in the same study, they measured VO2 max, which is an indicator of fitness, and they compared that to the size of the hippocampus. And kind of the bottom line of this is that there was a significant correlation such that individuals with higher VO2 max uh, overall had higher or larger size of the hippocampus. They also measured BDNF levels, brain-derived neurotrophic factor, and there was a correlation there too that individuals with higher aerobic fitness, VO2 max, had higher BDNF levels. There's been a lot of work done in rats and mice in particular, and some in humans, trying to understand what's going on in the brain that might uh, explain beneficial effects of exercise. So there's many animal studies showing that if you put running wheels in their cages, and then you have them compare them to animals with no running wheels in their cages, the animals that exercise every day perform better on tests of learning and memory, solving mazes, for example, shown here in the lower right, a depiction of a maze. And it's also been shown that the physical exercise increases blood flow to the brain, and actually over time, over periods of weeks and months, stimulates the growth of the blood vessels, which we call angiogenesis. So that's similar to what occurs in your muscle. When you exercise, there's increased blood flow, and then over time, uh, there's the proliferation of the vesicles or growth of the vessels so that you get more blood when you exercise again. Um, it's also been shown that aerobic exercise in rats and mice increases levels of brain-derived neurotrophic factor, this protein that we know plays a very important role in learning and memory. It promotes the growth of dendrites and formation of new synapses. And it can actually, and we were among the first to show this, BDNF can protect neurons against conditions that are occurring during normal aging and more so in neurodegenerative disorders, for example, oxidative stress, metabolic challenges, and so on. Another growth factor that's upregulated is fibroblast growth factor 2, which promotes neural survival and growth. And then VEGF, shown on the right here in the middle, vascular endothelial growth factor. Actually, that's what stimulates the growth of the blood vessels. Okay, so in animals anyway, rats and mice, aerobic exercise increases growth of dendrites, increases number of synapses, increases blood flow, and enhances learning and memory. Now, in the hippocampus, which is the brain region, plays a fundamental role in learning and memory, short-term memory, the establishment of memories from your experience. Um, Dr. Henriette von Prague, when she was at the Salk Institute, discovered that exercise increases the production of new neurons from stem cells, a process called neurogenesis in the hippocampus. And I've had a couple of podcasts that have focused on neurogenesis, and I kind of note those down here, Dr. Rand and Dr. Thurett. Okay, so that's pretty interesting. And then, so there's a lot going on in terms of the structure and function in response to exercise in the brain. The structure of neural circuits uh, is affected by exercise in good ways, and the function is affected. And also, I should point out that pretty much anybody who exercises knows this. Your mood is improved when you exercise regularly. And BDNF is thought to play a role in that. And there's been some work on that, looking at indicators of anxiety and kind of depression-like symptoms in animals. And the exercise reduces anxiety and lessens depression as well. Okay, recently, just in the last, well, it's been 10 years now, there's a lot of interest in what are called Exerkines. These are proteins or other molecules that are produced from one organ, and then in response to exercise, they're released into the blood, and they go to the brain and play roles in some of these processes I've talked about on the right here. So, for example, there's evidence that these three proteins, which are derived from muscle, cathepsin B, interleukin-6, and irisin in response to exercise, they can get into the brain and play roles in neurogenesis and synaptic plasticity, maybe learning and memory. Um, from the liver, beta hydroxybutyrate, uh, that's ketone body. Uh, important for energy metabolism, as I've talked about in previous presentations. It's used to produce ATP in mitochondria, but it also has signaling functions, which I'll talk about a little later. Um, Insulin-like growth factor 1, that's also produced in liver in response to exercise, and it uh, can promote hippocampal neurogenesis. And then your fat cells actually, in response to exercise, release a hormone called adiponectin, as well as this anti-inflammatory interleukin, interleukin 10. And there's evidence that by administering these to animals, that it can protect brain cells against stress and may even and and inflammation. This slide is from Henriette von Prague, who, after she um, worked at the Salk Institute, I recruited her to my laboratory at the National Institute on Aging, and she did a lot of really elegant studies where she used fluorescent probes to label specifically newly generated neurons. And then she also developed with group before at the Salk before she moved to Baltimore that enabled her to identify any neuron that forms a synapse with a newly generated neuron. So in this slide, these images here, the green is newly generated neurons. And again, if you're listening to this on Spotify or something, I'm showing slides on my YouTube channel. Uh, okay, anyway, so there are stem cells in the dentate gyrus of the hippocampus, this kind of C-shaped or backward C-shaped, as it's here, region. And shown here are each of these green neurons is one that had arisen from a stem cell during the course of the experiment, which was a month or so of physical exercise. And so you see there's a lot of newly generated neurons. They've grown dendrites and they've formed synaptic connections. They've received inputs from other neurons in the hippocampus, from neurons in other brain regions, for example, cholinergic neurons from the basal forebrain or uh, serotonergic neurons from the brainstem, etc. Okay, so when you compare, and this upper is just an example from a runner mouse. And then in this bottom schematic diagram is just showing that uh, compared to the control animals that are sedentary, the runners have more newly generated neurons, so there's more neurons shown here. And interestingly, uh, these new neurons receive inputs from other neurons that are already existing outside of the hippocampus. So there's this kind of increase in number of inputs coming into the dentate gyrus from other brain regions, and that's thought to play a very important role in a process called spatial pattern separation. And I'll show you, and this is very important from an evolutionary perspective because the hippocampus is very important for navigation through environments. And if you're like walking through the woods, it's important to remember what you see, where it is, not only in terms of getting back to your home after you've gone out in the woods, but also in knowing where different hazards are, whether it's if you're a prey animal, a predator, or there may be physical hazards, and remembering where they are. If you're a predator or a human, say hunter-gatherer, remembering where different sources of fruits, nuts, whatever, or where you're likely to find fish in the stream or deer, whatever. So anyway, the hippocampus is very important for that, and part of that you have to be able to in your mind and the neural networks in your brain have to be able to have a map, a cognitive map of where you are. So essentially, and this can be evaluated on a touchscreen. And this is some work from Dr. Von Prague when she was at NIA in my group. So she developed this touchscreen system. You can teach a mouse that if it touches an icon on a touchscreen with its nose, it will receive a food reward. And then you can teach it that you put two lit icons separated by a big distance, and it learns that if it touches this icon with its nose on the left here, it gets a food reward. If it touches one on the right, it doesn't. So these are separated by a big distance, and they learn that pretty well. But when you decrease the distance between these two icons so that they're close together, the animal makes more mistakes. It takes some more trials or attempts to be successful. And what Henriette's group found is that the animals that have been running and are in good shape, it takes them less time to figure out how to solve this when you have a closely separated icon. So they have better spatial pattern separation, and that was correlated with neurogenesis. So I mentioned the exerkines. This is a little repeat of this, but there's a few other growth factors added here. FGF21, fibroblast growth factor 21, also lactate. If you exercise vigorously, lactic acid is produced, and that gets into the blood, and it can be an energy source for neurons in the brain. It may also have signaling functions. So that's an active area of research. Okay, now I'm going to quickly go through a summary of some of the findings that have effects of physical exercise in humans and animals in models of various neurological disorders. So Alzheimer's disease, there's been just a few studies indicating that people in the early stages of Alzheimer's disease, their cognition benefits from exercise, their performance on cognitive tests. In animals, there's animal models of Alzheimer's disease. Physical exercise has beneficial effects there on their learning and memory, reducing their anxiety levels, and it may modify the amyloid and tau pathology. I mentioned anxiety and depression, a lot of work on that in both humans and animals. Autism, physical exercise is good for humans with autism. And, they often have trouble, they have kind of like an anxiety-like phenotype, right? They tend to avoid social interactions, and then they have these repetitive behaviors. And there's also animal models of autism where exercise has beneficial effects. Addiction. There's quite a few studies suggesting that, in fact, it's interesting, some big cities I've seen have running groups to help addicts get over their addiction. Depression, I mentioned. Epilepsy. There's pretty good evidence that regular physical exercise can help reduce the incidence of seizures in people with epilepsy. They have to avoid getting dehydrated. Huntington's disease, some benefits. Multiple sclerosis, definitely. Parkinson's disease, definitely. PTSD, definitely. Again, this is related somewhat to anxiety disorders, probably similar mechanisms. Later, I'll show you that the importance of GABA, the inhibitory neurotransmitter, in some of these effects. schizophrenia, stroke, um, substance abuse, I mentioned, and then traumatic brain injury. So exercise is good for your brain pretty much no matter what neurological disorder you have. Perhaps ALS, it's a little more murky. So that's the one neurodegenerative disorder, the motor neuron disease, Lou Gehrig's disease, where exercise is not so clear. Um, I'll just show you quickly some data on a protein that we've been very interested in, or had been uh, before I retired, and that is an enzyme called sirtuin-3 or SIRT3. And on the right in this slide, now, first of all, SIRT3 is located only in the mitochondria, and it's an enzyme that affects the function of various proteins by deacetylating, removing an acetyl group from the amino acid lysine in proteins. So a lot of proteins have lysine. Remember, there's 20 amino acids, whatever, methionine, glutamate, asparagine, et cetera, et cetera. But this enzyme removes acetyl group from lysine residues in proteins, and it affects their function in that way, similar to, I guess, what a phosphatase would do in proteins that are phosphorylated on certain amino acids. And then there are also acetyltransferases, enzymes that add the acetyl group to the lysine residues. Okay, in this study on the right, they identified about 100 proteins that are modified by SIRT3. They're deacetylated by SIRT3. Okay, so there's a lot of proteins. Some of them are involved in the electron transport chain. Some of them are involved in other aspects of bioenergetics, TCA cycle, etc. Some are involved in mitochondrial stress resistance and resilience. In this study that Awu Cheng in my lab was the main lead author on this that we published quite a while ago, 2016. What Awu did is we were able to obtain mice that lack SIRT3. So SIRT3 knockout mice, their gene has been deleted. So the mice don't have SIRT3. Now the mice seem fairly normal. We haven't done a lifespan study, but at least through middle age, they seem pretty normal. And however, when we stress them, they're vulnerable to injury and disease. And we actually looked at that in this paper as part of the paper. Uh, so in this paper, the first thing we did was ask the question, in the brain tissue, in the SIRT3 knockout mice, is there increased acetylation of proteins in the mitochondria? And we did that by a technique called immunoblot. And so we see all these different proteins here and the stronger intensity staining with the acetyl-lysine antibody. So we have an antibody that binds to proteins only when they're acetylated. And on the right here is isolated mitochondria. So yes, the SIRT3 knockout mice, there's increased acetylation of many proteins in the mitochondria. Okay, then what we did a couple of things. We did cell culture studies where we take neurons from embryonic mouse brain, cerebral cortical neurons, put them in culture, let them grow axons and dendrites and form connections. And then we ask a simple question. Are these neurons or are they not more vulnerable to stress? And we used an excitotoxic stress, either the excitatory neurotransmitter glutamate, which if you're interested, you can read about that important neurotransmitter in my book, Sculptor and Destroyer. It's the most important transmitter in the brain for our function. but there's also this process of excitotoxicity where neurons can be overstimulated by glutamate to the point that they degenerate and die. So the bottom line here is the red lines show the results with the mice lacking SIRT3. Many more neurons are killed and by lower concentrations if they lack SIRT3 compared to the neurons that have SIRT3. All right, and then we found that running wheel exercise increases SIRT3 protein levels in both the cerebral cortex and hippocampus of these mice. And so we quantified both the protein levels and the messenger RNA uh, of SIRT3. And then what we did is we asked the question, if we excite the neurons a little with very low levels of glutamate or other chemicals that activate glutamate receptors, NMDA in this example, does that have an effect on SIRT3? So the idea here was we expose the cultured neurons, this is cultured neurons, to very low levels and that don't kill the neurons. So these are kind of, we think, relevant to just increasing normal activity in neurons. And that increased SIRT3. So interpretation there is the mild exercise challenge, increased exercising of neurons within physiological limits increases SIRT3 protein levels. Okay, right. And then we found that in animals with running wheels in the cages, SIRT3 protein levels go up in both the cerebral cortex and hippocampus. Then finally, what we did is we asked the question, since we'd shown that increasing activity in neurons is sufficient to stimulate SIRT3 production, what about if we block glutamate receptors? If we decrease activity in neurons, will running still be able to increase SIRT3 levels? And the answer is no. We used a drug called MK1, which blocks these NMDA glutamate receptors, and that abolished the ability to exercise to increase SIRT3. Okay, and later on I'll show you that the SIRT3 is very important in protecting neurons against stress. So the rest of my presentation, I'm going to focus on intermittent fasting. This is a slide I used in a TED talk I gave 11 years ago. It was at Johns Hopkins TEDx talk, and it just points out that there's many famous intellectuals that claim that their brain worked better, they did more critical thinking better when they were in a fasted state. And then Mark Twain, also a very bright and witty guy, he says a little starvation can really do more for the average sick man than the best medicines and best doctors. And I think that's pretty much true now. Bioenergetic challenge, exercise and fasting intermittently, and keeping your nerve cells active like we're doing right now, me talking and you listening. Those are the three things that we know are good for your brain as you age and help you function better and have your brain more resilient as you age. Okay, so this slide shows what happens to glucose levels in the blood and ketone levels in the blood with three different eating patterns. The upper one is the typical American eating pattern: breakfast, lunch, dinner, and evening snack. Every time you eat, uh, um, your blood glucose levels go up, insulin levels go up, which then promotes removal of glucose from the blood, and then so the glucose levels go back down. But if you eat breakfast, lunch, and dinner, evening snack, so you're eating something during most of your waking hours, you won't even get an elevation in ketones unless you exercise. So for example, if you were to eat breakfast, lunch, and dinner, then get up in the morning and go for a run before you ate breakfast, probably your ketones are going to go up during that run. This second example is someone who does a complete fast 24 hours one day and then eats breakfast, lunch, and dinner the next day. And so on the fasting day, after about 10 to 12 hours, 10, 12, 14, somewhere in that time range, there's a metabolic switch to ketones. So the glycogen stores, which is glucose in the liver, is depleted. Fatty acids are released from the fat cells into the blood, and they're converted in the liver to the ketones, beta hydroxybutyrate and acetoacetate. And then, so during that fasting period, these many cells, and particularly excitable cells like your neurons, your cardiac cells, or your muscle cells, are using ketones as an energy source. This lower example here, which has become very popular eating pattern for people doing intermittent fasting, including myself, although I've done it for 40 years, and uh, uh, and now the general public just learned about this in the last 10 years. What kind of pioneered the early animal research with a lot with alternate day fasting and some with time restricted eating, but there's many others, including Sachin Panda, who I did a podcast with, who's done more than us on this time restricted eating. But anyway, in this case, the individual ate all of their food, like 3 meals, if you will, during a 6 hour time window. So they're fasting 18 hours every day. So every day they're having the metabolic switch and ketones are going up. Okay, there's a huge literature on intermittent fasting, animal studies, randomized controlled trials in human. This is an exploding area of human research now. There's, last time I checked, over 200 clinical trials of intermittent fasting in humans with various conditions. Still some, it's very effective in obesity, type 2 diabetes, but other conditions, cardiac conditions, um, and a lot of neurodegenerative disorders are neurological disorders as well. Okay, cancers is a big area. After I retired from the National Institutes of Health in 2019, I finally had time to write a book on the science of intermittent fasting. Uh, Even though we'd been doing research since the 1990s, I never had time to actually write a book. We just published the, scores of scientific studies over that time period. All right, so animal studies, rats and mice. The most common way until recently that intermittent fasting was done in rats and mice was every other day fasting. So 24 hours, no food. The next day, food available 24 hours. Keep repeating that every other day fasting. When that's initiated when the animals are young, they live, uh, in this study done in Worcester rats, they lived about twice as long as the animals that had food available and ate every day. And this lower graph here shows the body weights from these animals. And what you see is when the animals in the fed ad libitum every day, the controls, they gain a lot more weight than the animals on intermittent fasting, which you'd expect. And this is mostly fat, abdominal fat. The animals on every other day fasting, they maintain a body weight without this bad fat, but their muscle mass is pretty much similar, lean mass, to the animal's fat ad libitum. So the intermittent fasting is not adversely affecting muscle. And in fact, I'll show you at the end of my presentation that if you combine uh, every other day fasting with daily treadmill training in mice and let them go for 2 months, there's a additional benefit of doing the intermittent fasting in terms of physical endurance. But anyway, I'll show you that later. Also, I want to point out one interesting thing. Um, when you do severe calorie restriction or time-restricted feeding, which actually turn out to be the same in most studies, which I'll tell you later, the animals could lose a lot of body weight when you restrict their calorie intake. And if you restrict them to the point of starvation, the size of every organ except the brain and the testes in males decrease. So the brain is preserved during starvation. Okay, so it turns out that pretty much all of the published studies on caloric restriction in rats and mice are also daily time restricted eating studies. And the reason is the way the studies are done, the animals first determine how much each animal eats every day, and then the animals are divided into two groups. One group, they're allowed to eat ad libitum the same amount every day. The other group, they're given, in this case, 40% fewer calories than they normally would have, but their total food allotment is given all at once. And when they're calorie restricted, this is major calorie restriction like this, they eat all of their food within about a 4 to 6 hour time window. So they're actually fasting up to 20 hours a day. And their ketone levels go up, beta hydroxybutyrate, and their glucose levels are maintained low uh, during this. Okay, and then we were the first to show in this PNAS study, Mike Anson and others in the group, that some beneficial effects of intermittent fasting can be dissociated from any change in calorie intake. So we were able to show by manipulating fasting and calorie intake that we get beneficial effects in terms of improving blood glucose regulation, lower blood glucose, lower insulin levels, and increasing ketone levels in the animals on intermittent fasting compared to caloric restriction without fasting. This is moderate level calorie restriction. Okay, then a number of studies have shown that kind of similar to exercise, but not as profound, intermittent fasting can increase BDNF levels, promote neurogenesis, particularly the survival of newly generated neurons. It can enhance synaptic plasticity and learning and memory, and can enhance performance in cognitive tests, maze tests. Okay, here's an example of combining daily running wheel exercise with intermittent fasting, and this is a work of Alexa Stranahan when she was a graduate student in the lab. She's now a professor down at Georgia in Augusta. So what she did in this study, she used normal mice, wild-type mice, and diabetic mice, DB/DB mice. The DB/DB mice have a mutated leptin receptor. Leptin is a hormone that when you eat a meal, it's released from fat cells in your blood, and it goes to your brain and tells you you're getting full. Uh, you don't need to eat anymore. So these DB/DB mice can't respond to leptin, so they overeat and develop obesity and diabetes. Okay, so she took these mice and she divided them into four groups. The control, which is ad libitum feeding, sedentary, no running wheels, ad libitum feeding, running wheels, caloric restriction slash daily time restricted eating, sedentary, and then the caloric restriction with intermittent fasting, running wheels. And she let them go for 3 months. And then she removed one hippocampus. So obviously we euthanize the mice, we give them an overdose of anesthetic. Then she took out one hippocampus and she stained with Golgi stain, and so that she can visualize under the microscope postsynaptic densities on dendrites. So these perpendicular projections from the dendrite are essentially postsynaptic sites. So these dendritic spines are essentially a measure of synapse numbers. Okay. So one hippocampus, she's quantifying numbers of synapses. The other hippocampus, she measured BDNF levels, this very important neurotrophic factor. What she found in the normal mice, there was a significant increase in both the number of synapses and BDNF levels with both exercise and intermittent fasting. And then with the combination, there is even greater increase in number of synapses and BDNF. She found a similar thing in the diabetic animals, beneficial effects of both exercise and intermittent fasting. She also found that, uh, there were fewer synapses in the diabetic mice compared to the non-diabetic mice. Okay, so what's happening here with fasting and extended exercise? Get glycogen depletion from the liver, breakdown of triglycerides and diglycerides in your fat cells. So these are broken down to free fatty acids, these short carbon chain fatty acids, say 8, 10 carbons. But anyway, these free fatty acids are transported into the liver and converted to acetyl-CoA, and then the acetyl-CoA is, through enzymatic processes, converted to the two ketones, acetoacetate and BHB, beta-hydroxybutyrate. So these can be used as an energy source. In neurons, they're broken back down to acetyl-CoA, goes into the TCA cycle, then generate ATP in the mitochondria. However, this ketone, BHB, can also affect gene expression. This was shown by non-neuroscientists in other cell types, and a few genes were shown to be upregulated by this ketone. And my lab and another lab showed that BHB can also stimulate BDNF production, and we found that BDNF then can stimulate mitochondrial biogenesis, the production and increase in number of mitochondria in the cell. So here's data from the upper is graphs from the work of Cristina Morosi when she was a postdoc in my lab. She showed that in cultured neurons, this ketone, BHB, or we called it hydroxybutyrate here, it will increase BDNF levels in cultured neurons, but only when they're incubated in physiologically low, kind of low normal concentration of glucose, like 1 millimolar. When she incubated them in higher glucose, like diabetic, she didn't see an upregulation of BDNF. Okay, and she found both the messenger RNA and the protein levels increased. And then this other study in vivo done up at NYU, New York, they actually infused animals with the ketone and found that increased BDNF levels in the brain. Okay, so Steve Conan, who I did actually 2 podcasts with, he developed essentially radio-labeled acetoacetate and used it with PET imaging, positron emission tomography, so that he can, in living human patients, inject low amounts of this radioactive ketone into the blood. It gets into the brain, and it's can be taken up and used by neurons. And in this study, what he did, he used the radio-labeled ketone, acetoacetate, and then he also had radio-labeled glucose and 2-deoxyglucose. And so he had the people at the baseline before they went on a ketogenic diet, he did PET imaging. And when they're not on a ketogenic diet, the neurons in the brain are using mainly glucose. So you see a lot of red and yellow. So that's high glucose utilization. And but post-ketogenic diet, they use ketones, and their utilization of glucose goes down a lot. And there's a direct correlation between the amount of ketones in the blood and the amount of ketones that are being used by the neurons. And then what we did is we took beta hydroxybutyrate, a ketone, what's called a ketone ester, which was developed by a colleague down at NIH main campus, and we gave it in the food. We mixed it into the food of mice, and in particular, a mouse model of Alzheimer's disease that develops amyloid plaques, neurofibrillary tangle-like tau pathology, and as they get older, beyond a year of age, they start to develop cognitive impairment. And the bottom line is we showed if we give these mouse model of Alzheimer's disease, this ketone ester, for many, many months, it will ameliorate their cognitive deficits. So it had the beneficial effects. And there's now a clinical trial ongoing of this ketone ester in patients with early Alzheimer's disease. And that's being run by Dimitrios Kapagiannis, who was a neurologist in my group, and he's still at NIA performing a lot of clinical trials. Actually, I'll mention a clinical trial of intermittent fasting he did recently. Okay, so this BHB has signaling functions that can affect gene expression. I'm not going to go into details here. I'm actually working on a perspective article for a journal on metabolic switching and how that may benefit health. Okay, so the ketones affect expression of a lot of different genes involved in a lot of different processes, and that's shown here. Can affect the autonomic nervous system, can suppress inflammation. This ketone can. It affects lipid metabolism. And okay, so this slide here is very important from the standpoint of take-home message. So what do exercise and intermittent fasting have in common? What they have in common is cyclic metabolic switching. Okay, from a metabolic challenge to a recovery. So the challenge is the exercise or the fasting. The recovery is eating, resting, sleeping. And the switching back and forth over time, we think optimizes health. If one never subjects themselves to the metabolic challenge, they don't get the benefit. And on the other hand, it can be overdone. Fasting can be overdone, get to the point of starvation, and exercise can be overdone. Main problems are injury, but there is actually evidence that ultra distance runners who run like 100-mile race distances and train, several hundred miles a week, as they get in their 70s and 80s, they're maybe more prone to some problems. Okay, so what's shown here on the left is a 24-hour clock. So this is 6 a.m. on the top, 6 p.m. on the bottom. This green section here is a 6-hour time period where this individual or any person doing this is eating. So this is the eating period. Okay. And then after the eating period, there's what I call a pre-ketogenic fasting period. So I mentioned it takes like 12 hours to switch from glucose to ketones. Now, if you exercise during this time period, this pre-ketogenic period will be shortened and the ketogenic period would be extended in this example here. But anyway, this is a person that didn't exercise that's during this 24-hour period. And their ketones go up during the 6-hour period. So during the ketogenic period, there's a lot of changes that occur systemically and in the brain and cells, including in brain cells. Systemically, I mentioned these, FGF21, I mentioned adiponectin, Klotho, this kind of longevity protein that's being studied a lot. It goes up with both exercise and intermittent fasting, and it can do good things for the brain. Quite a few studies emerging. Ghrelin, very interesting. It's called the hunger hormone because it promotes hunger. It's a signal that's released after after fasting for a period, and it goes to your hypothalamus and makes you perceive that you're hungry. But it's also shown to have an anti-inflammatory effect, as does adiponectin. Cortisol is very interesting. So normally we think of elevated cortisol as being a bad thing, like chronic stress, which is true. But cortisol levels also go up with exercise and with fasting. However, we showed 20 years ago, Jaewon Lee, when he was in my lab at the University of Kentucky, before I moved to NIH, he showed that the way that brain cells respond to cortisol differs in animals that are on intermittent fasting compared to animals that are subjected to chronic, uncontrollable stress, bad stress. And so this is kind of interesting that, the way cells respond to cortisol can differ depending whether the stress that one experiences is ex maybe exercise, for sure fasting, versus bad chronic psychological stress. Okay, and then in the cells, during the fasting period, ketogenic period, antioxidant defenses are increased, DNA repair enzymes are upregulated, protein chaperones that prevent the accumulation of damaged proteins are upregulated, as well as autophagy. I've had a couple podcasts with David Rubenstein on autophagy, mitophagy, the removal of dysfunctional mitochondria. So that's what happens during the challenge period. And in this case, it's the fasting, but similar with exercise, actually. Then during the eating period, and another thing here is that during the fasting period, particularly towards the end in the ketogenic period, the mTOR pathway is down, the pathway that promotes the uptake of amino acids, protein synthesis, and the growth of cells. That's suppressed during fasting. Then during eating, mTOR is increased, and your cells take up amino acids, synthesize proteins. They generate new mitochondria, mitochondrial biogenesis, growth factors that are upregulated in response to the fasting or the exercise. They then promote the growth, in the case of neurons, formation of new synapses. That's probably occurring mainly during the eating period and the kind of energy replete non-ketogenic state. So the cycling over time, over periods of weeks, months, and years between a conserve resources stress resistance mode during the fasting and a growth and plasticity mode during the recovery period, eating, resting, sleeping, promote optimal health, including brain health. Okay, here's a study showing that intermittent fasting can enhance memory in mice independently of any reduction in calorie intake. So again, a dissociation between calorie intake and a beneficial effect of intermittent fasting on the brain. In that study, the same study, they showed that this kind of is called a longevity protein, Klotho, is increased in response to the intermittent fasting. And the ability, and it's also correlated with increased neurogenesis. So Klotho goes up, increased neurogenesis. And I had Dr. Dana DeBall, who's done a lot of work recently on Klotho, talk about her work on Klotho in the brain in one of my podcasts earlier this year. Okay, coming back to SIRT3. Before I left NIA, Yang Lu, a really good electrophysiologist, in my lab at NIA, a postdoc. He very technically skilled in inserting electrodes into individual neurons in the brain and recording their activity. And this study, uh, what Young did is we had animals either on intermittent fasting, every other day fasting or not, and then let them go for different time periods, 1 week or a month. And in this study, we had both wild type and SIRT3 deficient mice, SIRT3 knockout mice. So we have mice with or without SIRT3, and then they're either on ad libitum control, eating pattern, or every other day fasting. And then measured levels of SIRT3 protein in brain tissue, and found that by a month, at 1 week, there was a trend towards the increase SIRT3 protein levels. By a month, there's a robust increase in SIRT3 protein levels in hippocampus. So kind of similar to exercise in general, intermittent fasting can increase SIRT3 levels in brain cells. Then what we did is we test anxiety levels of the animals in a test called the elevated plus maze. The intermittent fasting after a month reduced anxiety levels in the wild type mice, but not in the SIRT3 deficient mice. So that's very interesting. There's a connection between mitochondria and anxiety. That is, if there's reduced levels of this mitochondrial enzyme, SIRT3, the ability of intermittent fasting to reduce anxiety is lessened or eliminated. And similarly, the ability of intermittent fasting to enhance memory in what's called a water maze test is impaired if SIRT3 is not present. Okay, so there seems to be a very important role for this mitochondrial protein, or either through some effect on the mitochondria and then a secondary effect on synaptic plasticity. And so Young looked at activity at synapses. So what he did is he put electrode in a glutamatergic neuron, a neuron that deploys glutamate as neurotransmitter, excitatory neuron in the hippocampus. And he recorded what's showing here. He's actually recording inhibition of that excitatory neuron by GABA, the inhibitory neurotransmitter. So he's looking at the release of GABA from a GABAergic interneuron, inhibitory neuron, on to the excitatory neuron. And so in the normal mice on intermittent fasting, so what we're looking here is these downward deflections here are, these are the recordings, and each of these downward deflections indicates GABA receptor channels opening, quieting, hyperpolarizing the neuron. So with intermittent fasting, there's a large increase in GABAergic control of the excitatory neurons. In the SIRT3 knockout mice, intermittent fasting is not effective in enhancing GABA. Now, this could be an explanation for why intermittent fasting reduces anxiety, because if you increase GABA, that's going to reduce anxiety. In fact, all the early drugs used to treat anxiety act, such as Valium, diazepam, act on GABA receptors to increase their opening of the chloride channel. Okay, and then finally, we also looked at something called long-term potentiation, which is a measure of strengthening of synapses in response to a previous input to that neural circuit. And bottom line is the intermittent fasting enhanced somewhat the strengthening of the synapses, which may explain beneficial effects on memory. So in summary here, fasting and exercise, and also cognitive stimulation, I mentioned this, activity in neural circuits, will, through these pathways involving calcium influx and upregulation of brain-derived neurotrophic factor will affect expression of genes that enhance the health and functionality of mitochondria, that promote mitochondrial biogenesis, also that enhance removal of defective mitochondria and potentially pathological proteins like tau, alpha-synuclein, etc. And also genes involved in synaptic plasticity, the growth of dendrites and axons, and so on. Okay, I want to finish up by talking about some human studies looking at either intermittent fasting itself or interventions relevant to intermittent fasting on the brain. So this is a study I mentioned from Dimitros Kapagiannis, had 40 men and women between the ages of 55 and 70, and they have obesity and insulin resistance. So they're pre-diabetic. They are at risk for cognitive impairment and Alzheimer's disease. Okay, and then they were randomly assigned to either 5-2 intermittent fasting, 2 days a week. They only ate 400 calories. It was given actually in a shake form that they drank. And then the other group was what's called the USDA Healthy Living Diet, where they're given advice for healthy diet composition and calorie intake. Okay, then before they were started on these two dietary interventions, and 2 months later, a whole battery of analyses were done. Obvious things like body weight, um, blood pressure, blood chemistry, insulin, glucose levels, indicators of glycemia. But then in the brain, a battery of cognitive tests looking at different aspects of learning and memory, something called executive function. Um, and then also their physical activity was monitored. And then we got cerebrospinal fluid from many of the subjects and measured a number of different things that are relevant to cognitive impairment and Alzheimer's disease. Okay, so here's some of the data, and I'll just, if you want to go to the paper, you can find this. It's freely available on PubMed. Just put in Kapagiannis uh, intermittent fasting, and it'll probably be the first one that comes up. Okay, the the participants in both interventions lost weight. The subjects in the intermittent fasting group lost a little bit and significantly more weight over the 2 months. Both groups had improvements in glucose regulation, but more so with intermittent fasting. As you might imagine, the ketone beta hydroxybutyrate was up a lot more in the subjects on the intermittent fasting. Cholesterol levels were down, again, more so with the intermittent fasting and LDL level. Okay, cognition. Um, Here's data from two different measures of executive function. And one of them is called dimensional set shifting, the ability, it's kind of like switching between tasks or multitasking, the ability to kind of quickly switch your attention from one task to another. This fluency factor is, it's kind of like its name implies, its ability to be fluid in your verbal uh, processing. And so there was significant improvement with the intermittent fasting, but not with a USDA healthy living diet and this executive function test, as well as in the memory tests. These are essentially, so verbal learning is essentially memorizing some words, list of whatever, 10 words, and then there's a time delay of sometime 10 minutes or something, and then you're asked to recall as many words. So there's intermittent fasting improved that. Okay, I'm almost done. I did want to point out that something I haven't talked about at all is the autonomic nervous system. I guess I did mention the word once. So this is a system, the so-called parasympathetic and sympathetic arms of the autonomic nervous system control functions of pretty much all of your bodily organs. Best studied is heart rate, your heart. So the parasympathetic component slows down heart rate. It uses acetylcholine as a neurotransmitter. And the sympathetic speeds up heart rate. It uses norepinephrine as a neurotransmitter. Of course, epinephrine is adrenaline that's released from the adrenal glands, and that's actually induced by the sympathetic nervous system. Okay, so we did some studies a long time ago in rats where we monitored 24/7 heart rate and blood pressure, and we showed that intermittent fasting, every other day fasting, slows resting heart rate and decreases blood pressure. Not immediately, it takes several weeks. And then by a month, there's a highly significant reduction in resting heart rate and blood pressure. And we obtained evidence there's actually a role for BDNF in this. And the BDNF is enhancing the release of acetylcholine. So it's increasing the activity of these parasympathetic neurons. And that is similar to what's been known previously for aerobic exercise. The data are just shown here with intermittent fasting and daily time restricted eating, reduce heart rate. And then when you go back to ad libitum feeding, within a couple weeks, the heart rate uh, and blood pressure goes back up. So this isn't like you can't just do intermittent fasting for a month and then expect they're going to continue to see the effects on these systems. And then last, what about combining exercise with fasting in terms of physical performance? There's a lot of activity in this area now, a lot of studies, some published combining intermittent fasting with exercise training, either resistance training or aerobic exercise. There's studies in elite cyclists, some runners, and so I'll just summarize the human studies. Resistance training, intermittent fasting is conducive to building muscle with the resistance training. And the studies that are published, it didn't seem like there was an additional benefit of the intermittent fasting. That is, didn't seem like the intermittent fasting is getting more muscle mass. There were short studies, only like a month or so or two. So we don't know about long term. The endurance studies, there's several studies suggesting that intermittent fasting during endurance training can actually enhance performance somewhat. Some of the studies, there was some reduction in body weight, which that may, if you're running and you have some even a few pounds reduction in body weight, that may help your performance because you're carrying a little less weight. On the other hand, we did some studies in mice where we combined daily treadmill training, and here's the treadmill training regimen. We 45 minutes on the treadmill, and then over a period, every few weeks over a period of a month, we increased either the running speed, the treadmill speed, or the incline angle. And we had animals that were either fed ad libitum or every other day fasting during 2 months of treadmill training. And at the end, we did a maximum endurance test, and we found that the animals that had been on intermittent fasting performed significantly better than those that were not. And yeah, and we just confirmed they're switching to using fats, ketones during the fasting day, and that their ketones are up. And actually, with running, very interesting. There's a dramatically bigger elevation in the ketones with the combination of fasting with running. So maybe, possibly, something people want to try if you exercise is arrange your schedule so you exercise at the end of the fasting period if you're doing time-restricted eating, and then see how that goes in terms of your performance. This is the last slide. I just wanted to point out that, and this gets the idea, and there's been very little work on it. I've showed you pretty much everything I've told you about combining intermittent fasting with exercise, but there are other challenges that people do that are stresses, physiologically normal stresses, that is, stresses we experience during evolution routinely. One is heat stress, which in light of global warming, that's becoming increasingly important. So sauna, there's some evidence that sauna is good for your health, right? Exposing yourself transiently. Again, it has to be transient exposure to heat or cold. And so I think there's a lot of room for work looking at this in terms of both normal health and vulnerability to disease. I didn't talk about disease at all in my next presentation. I'm going to talk about bioenergetics and neurological disorders with a focus on age-related neurodegenerative disorders, Alzheimer's, Parkinson's in particular, also stroke, and how what's going on in terms of metabolism and during normal aging, and then how exercise and fasting may impact on those processes. And there's also implications in our New England Journal of Medicine review article in 2019. Rafa DeCabo and I put a final illustration in there on how kind of our suggestion on how intermittent fasting might be integrated into medical education, medical practice, and lifestyle in general. Okay, thanks for your attention, and I'll see you next time, hopefully. If you're finding these interesting, do feel free to comment in the comment section on YouTube, or even send me an email if you want to at my Hopkins email address, which you can find easily on just Google my name. Goodbye.